So why do we need to develop AOP, which is Adverse Outcome Pathway in toxicology? So AOP in toxicology are very essential. So first, they can be used to have a better understanding of the biological mechanism after an exposure to a chemical. So for example, it can be an endocrine disruptor. Or it can be used for predictive toxicology. So for example, when you develop predictive models, it can be helpful to have an understanding about if an endocrine disruptor is toxic or not. Or you can use it as well to reduce the animal testing, because you can create some computational modeling, for example. So to create an AOP, we need a very high scientific expertise that will be complementary, because an AOP is a conceptual framework that integrates existing knowledge. So for example, we can integrate knowledge from different databases, from laboratory works, or from scientific literature. So for example, we can use tools that exist. So it can be tools based on artificial intelligence, such as the AOP L-Finder tools. And this tool will allow to screen automatically literature that exists and extract relevant knowledge to build an AOP. So we have different examples of AOPs that have been developed in Oberon. So these AOPs were developed by integration of results from the different partners. So in Oberon we have 11 partners. And these partners have developed targeted experimental and computational test methods. And these results were integrated to build AOPs. So for example, we have AOPs with a phthalate DEHP. So that's a compound that is used in plastic that you can find, for example, in food packaging. And these chemicals has been shown to initiate some increased lipid accumulation that can be connected to increased body mass index in population. Another example is with the forever chemical PFOA, which will initiate some molecular disturbance that may be connected to obesity in the population.
Well, I think that what we have learned from the in vitro work that we have done in Oberon, which was focused on liver, on pancreas and adipose tissue, was, well, first of all, we have implemented different in vitro cellular models for endocrine disrupting chemical for EDC screening. And thanks to that, we have been able to understand how different endocrine disrupting chemicals can interfere with glucose and lipid metabolism and what are the main metabolic endpoints affected. And that is really important because that helps to identify compounds that may interfere, that can disrupt normal metabolic function and contribute to the progression, the development of metabolic disorders like diabetes, obesity and liver chronic diseases. Also, I think we have gained knowledge into the mechanism in pancreas, in liver, in adipose tissue, underlying that association between the exposure to endocrine disrupting chemicals and the progression to those metabolic diseases. And also we have gained advance in developing effective assays for EDC testing, for endocrine disrupting chemical testing in terms of metabolic disorders, for example, for hepatic steatosis and pancreatic beta cell dysfunction, diabetes. Well, I think that right now the test that is the most advanced in Oberon for regulatory purpose is the steatosis assay in hepatic cells, which is able to predict if a compound can produce hepatic steatosis. And this method is based in the quantification of lipid droplet formation through automated image analysis. And indeed, it has been demonstrated to be a suitable tool for the identification of potential steatogenic environmental pollutants. And right now is under cross-validation process. And also in Oberon, we have been working in the development of a pancreatic beta cell assay, which allow us to determine whether a chemical is able to alter glucose-stimulated insulin secretion, which is the primary function of the pancreatic beta cell. Well, regarding the research gaps, probably one is the combined effects of the endocrine disrupting chemicals. I think we need more research to understand the combined effects of the EDCs and their interactions, and also the potential additive, synergistic or antagonistic effects of these compounds, because some of the tests are focused on individual chemicals, but we know that in the real life it's much more complex. And probably also other research gap is related to developmental and lifespan exposure. Most of the tests are focused on short exposure to endocrine disrupting chemicals in the cells or in adult animals. But I think we need more research to develop methods that better mimic those developmental and transgenerational effects. And probably we need also some more work in terms of refining and standardization of cell-based assays.
So, omics studies, diascetomics and metabolomics, and particularly the regression, provide a holistic overview of the biological changes induced by the tested endocrine disruptors in the Oberon project, like phthalates, bisphenols, and PFAS. The gene expression and metabolic profiles, in fact, gave us information about potential biomarkers of exposure and actually elucidated perturbated pathways that can be served as indirect or direct key events, and that can help us make a more accurate, more precise health risk assessment, and subsequently provide more targeted regulatory strategies for protecting the human health from exposure to these endocrine disruptor chemicals that are in everyday life products. So overall, among the multiply omics experiments we performed in different in vitro models like pancreatic, liver and adipose tissue cells models, or in vivo models like 5 days and 5 weeks post-fertilization zebrafish, we found consistently perturbed pathways related to lipids and amino acids metabolism, like sphygmolipid and rigorous phospholipid metabolism and tryptophan metabolism. That needs, of course, further investigation in order to be connected with the in vitro study results.
Differences in exposure level to endocrine disruptor can be explained by various factors. First, in terms of regulation, we know that the regulation is different across country, with the example of bisphenol A that was restricted before the other country, and so we observe a lower exposure level in French people than other European. Secondly, we also have different habits, so this can explain differences according to ages and sex, depending on the use of cosmetics or diet. And similarly, we can observe differences with age, again, because diet change over ages. Also because some contaminants bioaccumulate in the body, so if we measure exposure to DDE, for example, in blood, the level will be higher in older people than in young children.
Why are we using zebrafish to study obesity in humans? Because we have an inner fish, meaning that we have an ancestor, common ancestor with this animal, and there is a conservation, very high conservation of biological processes implicated in the storage of energy in adipose tissue. So the ZOT method is very highly operative It can be used by every laboratory because the protocol is very well defined. And in addition, it has been developed tools by using artificial intelligence for performing the analysis of the results. In fact, to decide if a chemical molecule or a cocktail of molecules can higher the obesity level, we use what we call the decision tree, meaning that we mix the epidemiological data with in vitro data, also in silico data, and at the end in vivo data. And it is what we are performing with in vivo data because it is a highly integrated model. For example, for endocrine disruptors of metabolism, you can have a chemical acting on the endocrine gland in the ability to synthesize, for example, hormones like insulin, for example. And if you impair this functionality, you impair also by an indirect effect the hormones, the tissues that are targeted by these hormones.
Classical pharmacokinetics focused on and is able to simulate the absorption, distribution, metabolism, and excretion of a chemical in the body. In addition, you have several parameters that can vary among populations, such as age, sex, body weight, and also exposure conditions such as the mode of administration, the mode of exposure, the level of exposure, and all of this variability could affect significantly the exposure of this population. In this context, PBPK models are able to take account of this variability by providing physiological parameters to this model, such as blood flow, cardiac output, organ weight, organ volumes, metabolic clearance. You can have a quite accurate prediction of the fate of the chemicals in general population, but also in subpopulations, such as sensitive population, infants, toddlers, transplant patients, elderlies, pregnant women. PBPK models are actually mathematical equations that summarize what is occurring in the body, a body that is consisting in several compartments, each compartment representing an organ, and each organ is connected with a liquid, mainly blood. Well, in the context of Oberon, we have to date developed 5 models so far. We are still developing some extra models. These models, for QSAR, these models are freely available on Vega platform. Vega is a platform initially developed by the Mario Negri Institute. It is made of it provides several computational tools in order to predict the chemical properties or activities of chemicals.